Classical treatments like surgery, chemotherapy, or radiotherapy targeted all the cancer cells without taking into account their environment. They can even be toxic to the surrounding cells, for example, in chemotherapy. Going back to a 20-year-old theory, researchers have started to investigate the possibility of using the patient's own immune system to fight back against cancer cells. The main theories behind the concept of immune therapy are called immunosurveillance and immunoediting, and rely on proven immunological facts. These theories were formulated in 1957 by McFarland Burnett and Lewis Thomas, who proposed that lymphocytes act as sentinels in recognizing and eliminating continuously arising nascent transformed cells. It has also been suggested that immunosurveillance primarily functions as a component of a more general process of cancer immunoediting, consisting of 3 phases. Every day in everyone's body, thousands of potential cancer cells are created: UV, radiation, smoking, but they don't reach a clinical status because the immune system recognizes these cells and eliminates them. This is called the elimination phase, where the immune system alone is enough to detect and eliminate before the potential tumor reaches a clinical threshold. This phase continues up to a point where a balance is reached and tumor cell proliferation equals the death caused by immunity. This phase is called equilibrium, and the tumor is still subclinical. The last phase of the process is called escape, where the tumor overwhelms the immune system and can't be stopped anymore. At this point, the tumor becomes clinical. In the next slides, we will briefly describe each phase and the mechanisms involved. During the elimination phase, the tumor cells release highly immunogenic antigens. These antigens activate antigen-presenting cells like macrophages, B cells, and dendritic cells. Once activated, these cells have 2 main roles. They activate natural killer cells, which then migrate to the tumor site and release molecules like granzyme B that will lyse with the tumor cells, thus releasing more antigens and feeding the loop. They also migrate to activate T cells by presenting the tumor antigens on their class II MHC, major histocompatibility complex, class II. These activated T cells migrate to the tumor where they release pro-inflammatory cytokines and proliferate. This combination of events leads to tumor cell death and elimination via classical pathways. During the equilibrium phase, tumor cell proliferation is perfectly balanced by cell death from the immune system response. The tumor does not grow but is still present and remains subclinical in most cases. There are many ways to explain the switch from equilibrium to escape. Once all the tumor cells expressing highly immunogenic antigens are eliminated, the only remaining tumor cells express and release poorly immunogenic antigens. The immune response is then lowered all the way through. Less activated APC, reduced migration, fewer activated T cells, resulting in less tumor cell death. On top of this, the tumor overexpresses immune checkpoint proteins and effectively blocks the activated T cells from killing it. Tumor cell proliferation overcomes the immune system and the tumor grows above clinical levels. Which are the tumor escape mechanisms? Tumor cells have a very high mutation rate, common to many tumor types. Loss of function mutations, affecting mainly apoptosis resistance, prevent the tumor from dying even if the apoptosis signal is received. BCL2 is a known example of this phenomenon. Another commonly modified pathway is survival, with gain and function mutations, for example in STAT3, that render the tumor cells independent from their environment. Simply put, tumor cells don't need a favorable extracellular environment to proliferate and are blind to cell death signals, thus enabling proliferation. Mutations also touch cytokine genes, and tumor cells start overexpressing suppressive cytokines like IL-6, IL-10, or TGF-beta. These cytokines can block the immune system and stop it from killing tumor cells. Tumor cells overexpress inhibitory checkpoints, thus effectively blocking T cells. Tumor antigens switch from highly to poorly immunogenic, and the subsequent activation of the immune system is lowered. Tumor cells also release molecules like PGE2 that can be highly immunosuppressive. Last but not least, tumor cells can attract suppressive cells like Treg or IL-10 secreting B cells. This phenomenon is even amplified by transformed fibroblasts surrounding the tumor, which release CCL2 and CXCL2, increasing the attraction. Treg, once at the tumor site, will act like an off switch on the immune system and turn off the response. The landscape of cancer therapies has been greatly modified following these discoveries. Passive therapies are immunology-based but don't engage the host immune system. They are also the oldest therapies, like monoclonal antibodies. Even though the public's eye has turned to immuno-oncology therapies, research still continues on passive therapies with multi-specific antibodies, oncolytic viruses, or cellular therapies like chimeric antigen receptor, CAR, T-cell receptors, TCRs, or tumor-infiltrating lymphocytes, TILs. Active therapies engage the patient's immune system in answering the tumor. Three main avenues are under investigation today: cytokine treatment with IL-2 or IFN gamma, therapeutic vaccines, immune oncology therapies combining checkpoint inhibitors or agonists of co-stimulatory checkpoints, or any combination depending on patient's background. Monoclonal antibodies were the first treatment tested and validated aside from classical therapies. Today, antibody treatments can be divided into two types: naked antibodies, meaning antibodies not linked to an active factor. These can activate apoptosis or inhibit tumor cell growth via their action on surface receptors. They can also be used to directly activate the complement or trigger ADCC, antibody-dependent cell-mediated cytotoxicity, via NK cells. Conjugated antibodies carry an active factor like a radioisotope, a toxin, or a specific drug. In this case, the antibody is used as a targeting tool to develop the effector molecule directly to the site of the tumor. Oncolytic viruses are a very new research area for oncology. The virus can be a non-modified virus that has, naturally, the benefit of only replicating in cancer cells because it expresses certain markers or proteins, or an engineered virus made to target cancer cells. In both cases, the end result is the same. After viral infection, the virus replicates in cancer cells. Virus release effectively kills the cancer cell and propagates the virus to the surrounding cancer cells. At the same time, cell death triggers cytokine release like GM-CSF and activates the immune system. Bi- or multi-specific antibodies have been studied for a long time. The aim of these projects can differ a lot depending on the company, so this slide is just an example of what can be done in an oncology context. In the example, the antibody is engineered to bring the tumor cell and the T cell into close proximity, while at the same time activating the natural killer cells. This antibody is designed to have a high affinity for the T cell receptor, TCR, and specifically activate this pathway, thus increasing cytokine release via the T cells. The tumor cell, being very close physically to the T cell, will immediately be surrounded by cytokines, thus making them more effective. At the same time, the Fc fragment designed to activate NK cells will mediate ADCC. CAR-T stands for chimeric antigen receptor T cells. These cells are engineered to express a fusion protein on their membrane. The extracellular fragment is a monoclonal antibody. This gives very high specificity and the benefit of recognizing the full antigen, while normal T cells only recognize the antigen when it has been digested by an APC and presented via an MHC. The intracellular fragment is equivalent to the intracellular part of the T cell receptor, and it activates the TCR pathway leading to cell death. When the AB side recognizes the antigen, it triggers the ZAP cascade and the cell releases toxic cytokines to kill the tumor cell. Today, we have reached the third generation of CAR-T, with variations on the intracellular side, different domains to activate one or multiple pathways with more efficiency. In therapy, blood is drawn from the patient and the T cells are isolated. The patient's tumor is sequenced to determine the best extracellular antibody sequence, and the construct is built. These cells are genetically modified and activated, then amplified before being re-injected into the patient. This means that each CAR-T is very patient-specific. since the construction is made to target this patient's particular tumor with high efficiency. TCR stands for T-cell receptor. As with CAR-T, we are looking at genetically engineered membrane receptors in cells. Contrary to CAR-T, here the whole structure remains similar to a classical T-cell receptor, but the extracellular domain is engineered to provide a better specificity to the tumor marker. Again, like CAR-T, the intracellular fragment is equivalent to the intracellular part of the T-cell receptor. It activates the TCR pathway, leading to cell death. T cells are isolated from the patient's blood, then they are tested versus the patient's tumor. The T cells with the best recognition are selected and the construct is made. Then cells are transfected and amplified. After activation, the cells are re-injected into the patient. Once again, this is a very personalized therapy, since each TCR will be specific to the patient's own tumor. TILs stand for tumor-infiltrating lymphocytes and represent a mix of T cells, B cells, and natural killer cells. There is no cell engineering in this therapy. TILs cells are present at the tumor site, are infiltrated or not in the tumor, and are highly activated and tumor-specific. In other words, they are the GIs of the immune response. In a TIL treatment, the cells are harvested from the tumor, cultured, expanded, then overactivated by IL-2 before being re-injected into the patient. Technically, it amounts to injecting highly trained and supercharged soldiers into the tumor for maximum effect. Even though this treatment is generally run in the same patient, it has been stipulated that it could be transferred from patient A to patient B if patient B, for any number of reasons, lacks a significant TIL population. Cytokine treatment is the first and the oldest of the active immune therapies. The treatment simply involves a perfusion of cytokines into the patient. As of now, the only FDA-approved treatment are the perfusion of interferon alpha to activate NK cells and induce tumor cell apoptosis, and IL-2, commonly known as the T-cell growth factor, which is classified as active since it directly activates the host immune system to attack and kill the cancer cells. Other possibilities are being studied, like GM-CSF, but none has yet received FDA approval. Therapeutic cancer vaccines, TCV, is a promising class of active immune therapies. TCV strengthen the immune response. The only FDA-approved vaccine is Sipuleucel for prostate cancer treatment. TCV differ from classic vaccines in the sense they are not preventive but therapeutic. The diagram here shows how Sipuleucel works. The dendritic cells, or APC, are isolated from the patient. They are genetically modified via viral infection, or now CRISPR, to express highly immunogenic antigens. Then they are re-injected into the patient. The cells, once back in the patient, are highly immunogenic and will trigger the immune response cascade by activating T cells and the other components of the response, leading to tumor cell death. Due to the effectiveness of two checkpoint inhibitor drugs for melanoma, ipilimumab and pembrolizumab, immune checkpoints are probably the most talked about of the active immune therapies, both within pharma and in the general public. They are co-receptors expressed naturally both on the T cell and the APC, antigen-presenting cell, and are normally used to modulate the immune response when the T cell receptor, TCR, recognizes its ligand on the major histocompatibility complex, MHC. Immune checkpoints are classified in two categories: stimulatory, inhibitory. If a stimulatory checkpoint like OX40 is activated, it will superactivate the ZAP pathway and activate other pathways, leading to an increase in cytokine release and generally increased T activity and lead to cell death. If an inhibitory checkpoint like PD-1 or CTLA-4 is activated at the same time, it will block the signaling cascade and prevent T cell activation.